Okay, welcome back to the last part of my talk. So I'm showing this slide again about my research. So we have two arms of our study of our research. One is identifying the genetic basis of cancers that we study. So these could be mutational signatures, population and cancer-specific driver mutations that we discussed in my previous part. And then we do some functional analysis, so different experiments to determine DNA repair defects, genomic instability, and then also screening for new drug targets. So the idea for doing this is to identify new or novel biomarkers that we can then use different genomic technologies for early detection of cancer, detection of minimal residual disease. So this is disease that is left over after treatment, and then also for targeted therapy. So for giving precision medicine or personalized medicine based on individual cancer patients. So some of my earlier work focused on human papillomaviral associated cancers. So HPV can cause different kinds of cancers. So HPV has been associated with almost 91% of all cervical cancers, anal cancers, vaginal, oropharyngeal cancers, and other anogenital cancers. So, so the way HPV causes cancer is by two mechanisms. One is by expression of this viral gene called as E6. E6 causes degradation of P53, so so that leads to cancer progression, and then also E7 that leads to degradation of PRB or retinoblastoma protein. That also causes overexpression of P16. So actually P16 overexpression is actually a surrogate marker for detection of HPV associated cancers. So there are different tests for looking at HPV as a biomarker for these cancers. So most of them, they use PCR-based techniques, P16 immunohistochemistry, which is a surrogate marker for E7 gene. And then HPV DNA PCR, this is qPCR based. This is actually pretty sensitive. And then HPV DNA or RNA in situ hybridization. So most of these have quick results, but unfortunately, most of these are not minimally invasive. So what I mean by that is that you always require tissue biopsy for taking these samples. And sometimes you also cannot get a measure of tumor burden from these samples. So what we thought was we'll develop a test so that is minimally invasive. So it's very hard that you call in a patient and always take a tissue biopsy, because getting a tissue biopsy is almost like poking a voodoo doll so many times. So it's the tissue, the cancer are not always accessible. And what we thought was that maybe we could develop a liquid biopsy test so we can try to get the same information of the cancer from a simple blood test. So ideally, we want to take the blood, spin the blood down to plasma and the rest of the blood. And then the plasma is a rich source of circulating free DNA or cell-free DNA. And then analyzing the cell-free DNA, we can use it for early detection or treatment monitoring. So what we do is, so initially what we planned to do was, this plasma is a rich source of cell-free DNA and also exosomal RNA. So first we started to see, to isolate exosomal RNA and cell-free DNA from plasma. So whenever we got a patient sample, we take the blood and spin it down for 800 G for 10 minutes. And then remove the supernatant, which is plasma. And then we spin the plasma again for 16,000 G times 10 minutes. And then we spin it again in an ultra centrifuge for 120,000 times for almost 1 hour. And then the supernatant will have circulating free DNA or cell-free DNA. We use this kit to extract the DNA. And then the pellet will have exosomal RNA. So they are exosomal RNA kits. And then these RNA also is a good source to look at microRNA and to see if we can use it for HPV microRNA detection also. So first we looked up, so we took one patient sample, and also we also took these cell lines called CHA cells. So we took these cells because this is a cell line where every cell has 1 or 2 copies of HPV integrated into the human genome. So we took these cells and we did a qPCR analysis to see whether we are able to detect exosomal RNA and cell-free DNA. So if you look at the yield of exosomal RNA is much lower compared to cell-free DNA in this. And then we also looked at the number of HPV copies, and then in these cell lines, we had a better yield of cell-free DNA compared to exosomal RNA. So we decided that it's better working with cell-free DNA for detection of HPV compared to exosomal RNA or DNA. So this is from a previous paper. So cell-free DNA is very, it has a signature of 150 to 200 base pairs. So because, when the cells come out of, so both healthy cells and cancer cells, when they die, they spit out genomic DNA into the blood. But the problem is during these apoptosis or senescence pathways, the DNA is being cut by these enzymes called as caspases. But as I told you in the first part of the talk, these DNA are bound by histone molecules. So wherever the DNA is bound by histone molecules, these caspases cannot cut the DNA. So that is what you basically see in the plasma. So the plasma cell-free DNA has a distinct 160 to 170 or 150 to 200 base pair fragments. So we are planning to take that and analyze for HPV or other targets of our interest. So there was another paper published in 2017 where these scientists, they took cell-free DNA from patients with nasopharyngeal carcinoma, and they were able to detect Epstein-Barr virus as a biomarker with pretty high sensitivities of 97 to 98%. So there are also other companies like Cobas that have already plasma-based liquid biopsy tests to look at non-small cell lung carcinoma patients for mutations in EGFR gene. So these are also some of previous data from other publications where they have found that the concentration of total cell-free DNA in the plasma is pretty high in these cancers. So these are head and neck squamous cell carcinomas that are also infected with HPV, mostly in the West. Not so much in India, or studies are not done in India to look at HPV prevalence in head and neck cancer. So here in this graph, you can see the concentration of cell-free DNA on the y-axis, and then you can see the patient. You can see the patient before therapy. These are patient samples before therapy and during therapy, and then these are different controls. So here there are 3 patient samples. One was taken before the therapy, one was taken after one therapy, and then after another therapy. So here you can see that before therapy, you can actually detect a lot of HPV-16. So these are CT values that correspond to qPCR. So just like you do COVID-19 testing by qPCR, this is a HPV-16 testing by qPCR. So the lower the value, it means that there is higher the virus in the sample. So here you can see that before therapy there is a higher viral load, and then during therapy there is a low viral load, and then after therapy there is not much viral load. So the sensitivity of detecting HPV by qPCR is very low, which is around 65 to 76%, which is pretty low. So we thought we should develop a test that is that is pretty high in sensitivities, and also that the frequency of cases with head and neck cancer with the detectable amount of ctDNA is around 65%. So we wanted to develop a much more sensitive test so that it is beneficial for diagnosis. So as I mentioned before, this is how a cell-free DNA looks like when you run it on a gel. So there's a mean peak of around 167 base pairs, but sometimes we also get this high molecular weight regions. So there are new technologies that are being developed that actually separate this particular fraction from the rest of the sample. Also, there are some technologies that want to separate circulating tumor DNA from the rest of the cell-free DNA. So what I mean by that is circulating tumor DNA is associated with the tumor, but the cell-free DNA actually is like a superset of all cell-free DNA that might come even from healthy cells or even from the tumor cells. So we looked up a lot of technologies that work on cell-free DNA and circulating tumor DNA. And we looked at these factors. One is that our test should have high sensitivity. There should not be a loss of starting material. There should not be amplification-induced bias, as we see in a lot of NGS-based assays that rely on PCR amplification. There should be a quick turnaround time. There should be tiling, so we should be able to do multiplex, and then obviously it should have low cost. So we analyzed a lot of technologies, and then what we came up was that droplet digital PCR or digital PCR technologies were actually pretty feasible to look up to make this liquid biopsy test. So as I mentioned in my previous talk, this is a droplet digital PCR workflow. So what we do is, in a typical PCR reaction, we have primers and template and buffers. But then this PCR reaction is being split into or compartmentalized into thousands of droplets. So in this reaction, we are splitting it into 20,000 droplets. This is a Biorad droplet digital PCR technology. So now every 20,000, so instead of having one PCR reaction, now we have 20,000 PCR reactions. And every PCR reaction, you can see whether there is an amplification of your target of interest or whether there's an amplification of the control gene that we have given. So then every droplet is analyzed for fluorescence. So here, if you can see, the blue fluorescence corresponds to HPV amplification. The green fluorescence corresponds to a control gene amplification. And then the orange is both, so that droplet has both blue and green, both HPV and the housekeeping gene. So now we analyze, we take a sum of these two, blue and orange, because that will give us the total of all the HPV in the cell, in the sample. So we first went on to do a primer design. So we analyzed, we made two primers for HPV 16, which is the most commonly found HPV subtype in both cervical and oral cancers. So we analyzed around 450 whole genomes of HPV 16 from this resource. And then we try to design primers that account for all the subtypes. So there are different subtypes. So there could be an African subtype, Asian subtype, European subtype. We try to account for all the subtypes and design these primers. And then what we did was we collected blood samples of around 100 patients. And for every patient, we did around 10 samples. So we collect them before treatment. So that is called a CRT. So that is chemoradiation therapy. So we collect a patient sample before treatment, during every step of the treatment, and post-treatment. So what we see that, so as I told you here, so the blue corresponds to HPV and the green corresponds to a housekeeping gene. So here you can see that, so this is the level of HPV before the treatment. And after every round of treatment, you can see that the HPV levels come down. And by the end of treatment, you don't see HPV at all. But here you can see the housekeeping gene, which is the EIF2C1 that has been maintained throughout. So this is like a positive control. So this is there in every sample, but then HPV levels actually decrease after every round of treatment. So we did some more standardization. We compared our ddPCR with other commercially available and qPCR assays. So here you can see the comparison between Droplet PCR and COBAS. And you can see that the digital PCR actually performed much better. And when you compare qPCR to digital PCR, you can see that the digital PCR, which is here on the y-axis, it actually compared better compared to the qPCR. But when you go towards higher viral loads, actually digital qPCR performed better. But we are more interested in early detection, so we assume that most of the samples will have very low tumor burden. So we are more interested in detecting very highly sensitive samples. So this is the data from our study from our 97 patients. So we had around 20 normal subjects. We couldn't detect any HPV-16 in these. And then we took some HPV negative cancers. So these are breast cancers that are not usually associated with HPV infections. So even here we couldn't detect it. And then in our 97 patients with head and neck cancers, we were able to detect HPV in 90 of them. And then there were 7 of them where we could not detect HPV in these samples. And then after surgery, we also again tried to detect them, but then we could detect any. And then we also had 8 anal cancer samples, out of which we could detect HPV in 7 of them. So then what we did was we took these 7 samples that were negative for HPV-16, and then we tried to measure HPV-33, which is the next most prevalent HPV subtype in head and neck cancers. So here again, in 20 normal subjects, the HPV-33 was undetectable. And in 7 of them, we were able to detect HPV-33 in 3 of them. So now if you add HPV-16 and 33, so on the y-axis, we have HPV-16 and 33 copies. And then on the and then in our 97 patients, we were able to detect HPV in 93 of them. So we actually developed a pretty sensitive technique that was able to detect HPV in 96% of these patients. So there was a sensitivity of around 96% and a specificity of around 100%. So, and then what we did was we analyzed for HPV in after every round of chemoradiation therapy. So what we found was in our samples, the HPV levels go down, and by week 3 or 4, it actually goes down to undetectable levels. So we think that this test could be used for early detection of HPV-associated tumors from a simple blood test. And then, so we wanted to really show that this test is useful in determining the cancers that are earlier, like at an earlier onset of cancers. We can actually use this test. So we used samples that had very low tumors, or these are samples that have resected where the surgery has been done to remove most of the tumor, but some of the tumor has been left out. So if you can see in these 3 tumors, there was the tumor was actually resected, and then we were able to still detect HPV in some of these tumors. And then this 4 and 5, they are intact primary tumors. So the tumors were intact, and we are able to detect them in, using our test. So we think that our test is pretty sensitive, even when there is a low burden of the tumor in the sample. And then we also measured our test along with other tests that include other driver mutations. So for example, in this patient had another mutation in the gene called KRAS G12D. So we used our liquid biopsy test to on the cell-free DNA sample to look for HPV and KRAS. And you can see that HPV was a little more sensitive compared to KRAS. There was another patient with a PIK3CA mutation, E545K. Even in this, HPV16 kind of performed a little better. But a lot more work has to be done to really show that this HPV16 is a better biomarker compared to the driver mutations. So what we think is that this, to summarize, our test is pretty sensitive and can be used for screening of cancers at high risk. And then we also think that this can be used for treatment response in clinical trials, such as radiation dose de-escalation. So if you see that HPV is gone, then maybe you can reduce that radiation dose. And then maybe to see detection of minimal residual disease after surgeries to inform clinicians about other therapies that they can use. So this is the test that we had designed, and then we hope to use it more at a diagnostic setting. So I'm going to move into another project that we are doing. This is a project in cervical cancer. So cervical cancer is a huge problem in India, and 91% of all cervical cancers are caused by human papillomavirus infections. And out of these, 70% is contributed by HPV type 16 and 18. So every year, almost 1 lakh new cases are diagnosed, and then 60,000 reports are reported annually. And actually, India only reports 1/3 of all global cervical cancer deaths. And there's a huge burden on the government and also on the healthcare system because of this, because these cancers usually are diagnosed much later on, so much later stages, compared to the West, where these women are subjected to, they get pap smears or examinations every 2 years. So you can actually detect cancers much early on in the West, but in India, these screening programs are not that common. So the cancers that we find are much later on, late onset. So as you can see here, the 5-year survival of patients with stage 1A disease is around almost 96%. So if you can detect cancer at a much early on stage, it is 96%. But if it is stage 4, then the survival is only 5%. So that is what we mainly see in India. So the accuracy for predicting relapse at, sorry, just move this. So here in stage 1B patients, they receive brachytherapy. The survival is around 94%, and as the stages increase, the survival decreases. And the accuracy for predicting relapse at 18 months is actually, if you take plasma, it is much higher. It is much higher at around 77% compared to just imaging, which is 60%. And patients with undetectable plasma HPV DNA at the end of radiation therapy had higher progression-free survival compared to those with detectable plasma levels. So if you see there is a detectable, if you detect HPV after treatment, these patients actually tend to fare much worse compared to undetectable level of HPV DNA. So there is a scope for using liquid biopsy tests during treatment and after treatment for detection of cervical cancer, and to see how these patients respond to therapy and if additional therapy has to be given. So we have embarked on a study in cervical cancer. So I have a PhD student who's working on this. So we recruit cervical cancer patients, and we take blood samples and tissue samples at every stage. So before treatment of the when a cervical cancer patient comes into our hospital, we try to take a tissue biopsy and one blood sample. And then from then on, we only take blood samples. So the next sample will be after EBRT, which is radiation therapy. And then the other sample would be again after brachytherapy, which is also a kind of radiation therapy that is given to some patients. And then we follow these patients for 3 months, 6 months, 1 year, and then if possible after 2 years. So this is how we collect the samples. So this is there are different stages. So we take if it is stage 1A or 1B, we take pre-treatment samples after surgery and after radiation. If it is any of these other stages, then we take pre-treatment after radiation and after brachytherapy. And then if it is metastatic, we only take pre-treatment. And then we follow up these patients for 3, 6, 12, and 24 months. And then we give the blood sample for HPV detection by droplet digital PCR. And then we also do whole genome sequencing on the tissue biopsy to actually really know whether this has HPV positivity, and also whether there are other driver mutations that occur in the sample. So as of now, we have collected a few patients. So these are all the patients that we have collected. I believe it's around 30 to 35 patients. And these are all the different stages of patients that we have collected. Most of them who come to our center are at stage 2. So this is the detailed methodology. So we collect blood, spin it, transfer the plasma, and then spin it again, and transfer the plasma and store it at minus 80 degrees till we further use it. And then we extract cell-free DNA using QIAGEN circulating free nucleic acid kit. And then we subject these samples to droplet digital PCR. So there's still a lot of work to be done, and we hope to follow up these patients, do the droplet digital PCR on these patients. We currently have some data from whole genome sequencing of our patients, and we are actually currently analyzing that. So with that, thank you all for your patience and listening to my talk. So this is my team. I have 2 PhD students, Preeti and Rama. Preeti works on the cervical cancer project that I was talking about, and Rama, he works on a head and neck cancer project that I'm working on. I also have a medical student, Kushbu, who is aiding us in manuscript preparation and also some experiments. I would also like to thank DBT for funding. Especially through the Ramalinga Swami Fellowship that I got in 2019, and it's a 5-year grant. So we have some funding for these studies. I would also like to thank the rest of the Department of Medical Genetics, the Faculty of Medical Genetics, faculty at Department of Radiation Oncology at MAHE, and Department of Pharmacy Practice. And my previous work that I showed you with the head and neck cancer project that was done in the Department of Radiation Oncology at Sloan Kettering Cancer Center. Thank you. And if you have any questions, you can always reach out to me by email, which is rama.damerla@manipal.edu. Thank you.